Hi, everybody. This is Cynthia Cleto. I lead the outreach and entrepreneurship efforts at MSIP. I hope everyone is recovering from the heat wave and enjoying the cooler summer weather. It's still summer, so please enjoy it as much as you can. Today, I'm excited to be here with Dr. Yasmin Hurd, Ward Coleman Chair of Translational Neuroscience and the Director of the Addiction Institute at Mount Sinai. We're big fans of Dr. Hurd here at MSIP and look forward to chatting about her experiences in work, life, and beyond. Hi, Dr. Hurd. How's your summer thus far?
Hi, Cynthia. It's been hot, but I have to say I'm probably one of the few people who really loves the heat. I'm always having debates with one of my postdocs who is the complete opposite. So this is to Jackie Ferland. I love it, but I even for me it did get a little overboard.
It did, but I'm glad we all survived it.
Yep.
So let's dive right in. We want to get to know you, Dr. Hurd. Tell us a little bit about your background and how did you know you wanted to pursue a career in neuroscience? And when did you start in Mount Sinai?
Yeah, it's definitely been a journey, let's just say that. So perhaps, with the heat, my journey began in Jamaica. So I love the heat, even though Jamaica doesn't get like this, like kind of the humidity. I mean, like, an island, tropical breezes are always there. But I started my journey to neuroscience because I was a weird kid. I always tell people that I was a weird kid. I always wanted to know why why certain people acted in a certain way. And even when I was really young, I knew it related to the brain. And so I was fascinated by the brain even back then. And it's not that I come from a family of scientists or physicians. So I say I was always weird. And then my family, we came to New York around when I was about 10 or so. And it was a interesting time. I love New York, from a small island to a city where I would go like this and you could hear the noise, and I
That's
was
so
just like,
interesting because it's so contrasting, the
exactly,
quiet of the island and then all of a sudden New York. Yeah.
exactly. So much so that I remember I was in a conference in a small island, and in the morning I was like, oh my God, all that noise woke me up. And they were like, what noise? I'm like, the birds. And they were like, oh, you New Yorker. So it's like, yeah, so I
Yeah.
was I was made for New York. So coming to New York was amazing, amazing. And it gave opportunities, obviously, in learning about science in a certain way, but it also brought challenges of being a Black girl that liked science, came from the islands, and, to kind of like, what do you do with her? So, that was the beginning, of my journey. And never in my wildest dreams would I have thought that my journey would take me to Sweden. And that's where I did my doctorate, and I was a professor also with a stop at NIH for my postdoc, and where I started my independent research lab. But it's, you never know where life is going to take you. So Jamaica, New York, Stockholm, and then to Mount Sinai. And it's crazy. I came to New York, Mount Sinai in, I hate to say the year because now it's like, oh my God, I'm old. 2006, but it feels like yesterday.
Mm.
And I remembered interviewing even with Dennis Charney and leaving his office and being on air thinking, this is the place that all of the things I want to do can be done, and it has been true. Definitely the translational work that I wanted to do, I was able to do here.
That's great. And we're so glad you're here and working on important research. One question I had was what sparked the idea for helping patients with addiction specifically?
You know, it's funny. My closest friends will tell you that, if they would have thought of any field in neuroscience that I would go into, it would not be addiction, because they used to tease me that I didn't even drink alcohol before I went to college. And, I complained about there not being non-alcoholic beverages when they had college parties. Back then, you were allowed to drink at 18. And, so I got into being fascinated by the neuroscience of addiction by my interest in studying neurodegenerative disorders like Parkinson's disease.
Mm.
And in Parkinson's disease, the cells that are lesioned are these dopaminergic cells. And these dopamine, as many people perhaps in the audience knows, a neurotransmitter that's really critical for motor function, but is also critical for reward, attention, for many different things. But in Parkinson's disease, we used to use pharmacological approaches to really tease out that system. That was before we had all these very fancy molecular techniques that we do now, that we use now. And some of the pharmacological approaches that we used were drugs such as amphetamine and cocaine, because these work by elevating dopamine levels by targeting this protein that brings the dopamine back into the cells.
Mm-hmm.
And so when we started using, giving these drugs to animals and so on, and started to understand more of cocaine and amphetamine, I was just fascinated by how much it changed behavior.
Mm.
And it was that kind of exploration that brought me to the field of addiction. So it wasn't because, I knew people at that time that had a substance use disorder. Of course, later on, I did, and I still do. And it was the neuroscience of it. It was just fascinating indeed how these drugs were so potent in changing behavior. And behavior, as I said, from even a kid, I was just fascinated by how, why people act a certain way, and that these drugs could really take over and even change certain fundamental behaviors. To me, that was fascinating.
Mm-hmm. And back then, what did you notice about the field of addictions, like back then when you started versus now? Like, how has it changed, evolved? Maybe what are some of the opportunities now and some of the challenges?
The field, like many others, has changed dramatically. Like I said, for example, even though we still obviously use pharmacological agents are still the primary thing. And for me, a huge part of the work that I've always wanted to do was to have research that impacted the human condition in developing some sort of treatment. So back then we used pharmacological agents. Now to tease out like neural circuits and specific places in the brain that it's critical for addiction related behaviors. And now we can use genetic tools. We can change the firing activity of cells in specific circuits in the brain. We can do very fine cell specific manipulations that weren't as finesse back then.
Mm.
Also, the field has grown dramatically. I think the neuroscience of addiction, we've learned a lot. As I mentioned earlier, when I first started studying dopamine, it was related to Parkinson's disease and its impact on motor behavior. And then we did know that increasing dopamine was associated with reward. And many people in the addiction field was, addiction is a disorder of reward, and therefore dopamine was the primary focus.
Mm-hmm.
However, um, computational neuroscientists started to show us that dopamine really was about the prediction of reward,
Mm.
and how the dopamine cell firing was about, as I said, predicting the reward. And as we studied more about the molecular changes occurring in the brain with addiction, we realized that dopamine may start the process, but it's all these other transmitter systems. And one of them, for example, our excitatory neurotransmitter like glutamate, which is important in the cortex, prefrontal cognitive function. And transmitters that really relate to and certain circuits in the brain related to like negative aversive states that
Mm.
really drive that cycle of addiction. So I think that the focus of neuroscience today is much more diverse than it was when I started studying addiction. And I think that's really great because it reflects truly the complexity of addiction, substance use disorders. And in general, it really reflects the complexity of most psychiatric disorders. And I think that that's the thing that, we all grapple with. But the one thing that I think um, I didn't appreciate back then, and as my career developed, the stigma of addiction
Mm.
is really huge, and I think unfortunately is still here today.
Mhm. And so it sounds like in its complexity, it's the nuances and subtleties are like slowly, like it's being built up and worked out. So the full spectrum is more at hand than it was, let's say, 15 years ago.
Absolutely,
Yeah.
absolutely, and that's a great way of framing it. That's exactly it, and that's exciting.
Yeah, that's very exciting because the minute thing we knew and thought of is now it has more color. And I'm wondering, how are you using what you know
Mhm,
from those subtleties and nuances to,
mhm.
work on your research? What is your research about, and how are you applying that fine lens onto your research?
So one of the things I decided to do, or it's not that I decided to do, it's just me and I think each of us where we even if we ask the same question, we go about it differently. And for me, a huge part of my research, as I said earlier, had to really deal with the human condition and translating it. So, were our animal models really reflecting what was happening in the human brain? So, I made a decision, even though it was very challenging and we definitely couldn't get our papers published at that time. I decided to study the human brain and look at it on a molecular level. And people tell me that, when we look at the expression of genes, for example, um, obviously it's postmortem, people have died, and, they're not stable, those particular molecules in the brain, everyone said. So it was very tough for people to believe the changes that we saw, but the
Mm.
changes actually were reflected in our animal models. So for me, at least it made me feel that the animal models that we were working with could help to inform us about what was happening in the human brain. Now, everybody's studying the human. We understand that we can conduct all these molecular studies in the human brain. And so that has opened up insights for many, many disorders. So when we saw these changes, it actually opened my eyes up because for me, it was clear that dopamine wasn't the driving force. It's still an important neurotransmitter in addiction, don't get me wrong. But now we could start focusing on other transmitter systems and other neural circuits. And so the question is, can we ever reverse these molecular changes that we see in the human brain? Can we come up with treatments that may even target certain circuits, certain cells that we can now develop really novel treatments? And so a big part of the work that I wanted to do was to understand more and more about those cell specific changes. And those are some things that my team, we're working on now more than ever. But it really is about what's happening in the human brain. Can we replicate that in the animal model? Can we then use the animal model to develop new treatments that we can then go back to humans in clinical studies and see whether or not they may be effective?
Mhm. And the most recent work that you're doing is in CBD, correct?
Yes. So, and actually, that again was a very long journey, and that journey actually started. I realized that's already. That journey started with the. I have two main big umbrella programs in my lab. One relates to looking at the neurobiology of opioid addiction, and the other looks at the developmental effects of cannabis. And when you look at the developmental effects of cannabis, we can study um, we mainly study animal models, and obviously people are not dying from cannabis overdoses like they are from opioids. So our brain bank collection obviously has more opioid people who died from opioid overdose. So we had really focused a lot on our animal models there. And when we study cannabis in our animal models, we often study THC. THC is the psychoactive cannabinoid in the plant that induces the reward, the euphoria. And we would invariably see in our animal models that those animals, whether prenatal or adolescent exposure to THC, would, for example, self administer more heroin when they were adults. So, and it's this issue of, and we can get into cannabis questions later, but when we study these animal models with THC, it's important to understand that the cannabis plant has over 140 cannabinoids. So THC is just one of them. And so we wanted to see, for me, it was like, is it really THC that's inducing this, or is it something else? And so we started studying another cannabinoid, and that's how we started studying cannabidiol, CBD. And back then, it was over a decade ago. No one was studying this. And we saw something unusual. We saw that in contrast to what we saw with THC, when we gave animals CBD, they would decrease their heroin seeking behavior. So it was, in a way, I either expected nothing or opposite to what we had expected when compared to THC. So clearly, the cannabinoids in the cannabis plant do different things in regard to addiction vulnerability and even psychiatric vulnerability. So then, for me, I wanted to see, we come up with all these ideas based on our animal models, but a lot of times they don't translate to the human condition. And so a lot of treatments that are really needed for psychiatric disorders have really not made it past any stage, because when they go into humans, they don't work. So I wanted to see if CBD did work before going into more mechanistic understanding of why CBD may be working and differently from THC. So we conducted human pilot studies, clinical trials, and they actually replicated what we saw in our animal models. So when we gave people who had an opioid use disorder CBD, it reduced their craving and actually also reduced their anxiety, but specifically with environmental cues. And that's one of the things about addiction that many people don't realize. You know, most people who suffer from these disorders, they do not want to be trapped in this, in a way you can say this hell. They're trapped because the craving, the urges. My God, I can't even stop taking a potato chip. And when I say no, I'm not going to eat this anymore. And these people, it's much harder. And this is the environmental triggers, the stresses that triggers these craving. And so that's what CBD, we saw that it was reducing the impact of these environmental cues to trigger the anxiety, to trigger the craving. And that became a whole path for us in terms of developing CBD as a potential treatment for opiate use disorders and certain other related psychiatric disorders.
And the treatment that is administered is very heavily controlled and dosed throughout the whole thing. So it's very specific, right, for the trial.
Yes. So, since we started this research, sadly, I mean, sadly and important, and I think it's also good. So, on one hand, um, cannabis today is now, legal in many states in the U.S., still not federally. And I think that that was important because people were getting arrested for using cannabis and destroying a lot of communities, and especially Black and Brown people, which to me was horrific. But we now have this thing that everybody thinks that cannabis or CBD can cure everything. And that's also, going the other extent as well, which is silly. So for clinical research and for medicine you have to do in a very structured manner. You have to develop in a structured manner. You have to know the dose. You have to know who may be, best suited for it. No one medication is going to cure every single thing for these complex disorders that have different phenotypes. And so it's important for us to understand who it can be better suited for, the dosing, and even the route of administration. So those are things that, we're in the process of trying to develop a much more structured path towards FDA approved medicinal use of CBD. But today we have CBD you can buy everywhere. It's in your coffee, it's in your hamburger, but that's not medicine. And I think that's really important for people to understand.
Another question I had was, how did your path cross with MSIPs? How did it come to be that our groups work together?
Well, it started with CBD, and it started with, I think Sinai, I will be completely honest, I never thought about patents or things like that. It was just like, okay, how can the work, our research really impact human lives? And we would publish whatever data that we obtained. And I remembered, I can't remember who sent out the email, about, and this about, think about, the impact of your work and in patent and so on. And so that's how it started. Was so like nearly 13 years ago when we got some of the first results from CBD. I was like, I wonder, could a cannabinoid, be developed as a medicine that could be relevant to some aspect of patenting. So that's how it started with MSIP. And I also the research that I do with other, it's not just only CBD that our research has trying to develop new medications. It's based on, like I said, results that we obtain from the human brain. There are other strategies, other ideas that we have. And so that relationship with MSIPs there. And in addition, what we've learned about the developmental effects of cannabis. So working with them, I'm horrible. All the legal things go Zoom. So that's why I reach out to MSIP and what we can do and what we can't do. And unfortunately, CBD, even though, I loved it because it was so unexpected, and it brought us down this path that we could potentially indeed develop a medication that could work in humans. When CBD changed in society, all of these companies, um, there are a lot of bad actors out there, and everybody wanted to make money on CBD. So working with MSIP, they have been able to, it's been, a struggle. Everybody wants to work with me, all these companies, but they also come with certain complications. And MSIP has been really great in helping to navigate how do you work with certain companies and which companies do you work with and which ones, should try to stay away from. And so they also help in that regard.
Great. We're there for you, Dr. Hurd, and looking
Thank you.
forward to the results of the work. Um, Perhaps since you brought up potato chips, and it's something that also in this era of staying home a lot, maybe not being able to see all the people I want to see personally, there are habits um, that I entertain, such as binge watching TV, potato chips, things like that. That isn't, an addiction the same way an opioid addiction would be, but it's kind of around that area. And I'm wondering, from the insights that you've gained in the decades that you've worked in this field, what are some insights that a layman, can kind of apply to simple things that I might have a slight addiction to, like my iPhone, for example?
Yeah, yeah. I mean, COVID has been horrible on so many levels, over 1 million people dead, and the change in our lifestyle and the change in our lives, and like you said, more isolation. And we did see that during COVID, more people have, abused alcohol who never even did before, and other substances. So, and as you say, the binge watching, the different, non-substance use, quote unquote, addictions have definitely also increased. And it comes back to the aspect of stress and isolation. So isolation definitely increases stress. And, for young people, this is particularly a tough time. So they rely a lot more on their digital technology, but it's not true connectivity. And this is one thing that I think we have to work on more. And it's so, for example, I said technology can also help. So instead of like binge watching TV, aspects of physical exercise, even though I'm not great on that myself, but perhaps Zoom with friends doing, a fun activity is better than doing it alone and just sitting on the couch, binge eating and so on. So there are things that we can still leverage the technology that could still give us greater social connection that would decrease our stress. And and that social connection and the decreased stress will decrease our this habitual behavior, especially for things that are not necessarily good for us, whether for our brains or our bodies as we, gain more weight, eating all the bad things that we have done during COVID, but also, it's how do you start reaching out and having true connections with people and with the people who you care for and care for you. I think that that's one of the things in terms of just making those types of connections more. But everybody has been facing it, and that is the thing. And the good thing is that we are quote unquote coming out of it in that, like the CDC just, um, changed the policies of, now you can be around people without your mask on in a certain way. And so I think it's real, and it's still summer, so at least here for us, spend time, go out in the park, meet friends, in the park, do things. And I think that we can try and break out of this so that we don't get into this habitual behavior. And it's also important to be a true friend. Addiction doesn't just come out of the blue. There's a process, there are stages. When you see someone getting into a pattern that you see may lead to something negative, it's important to start saying it early so then people start being more aware of what's happening to them. A lot of times we are very afraid, and I think sometimes even parents are very afraid to talk to their kids when they do see something happening, and they will like, well, not my kid because he's so good and I know he'll come out of this. But we wait until someone gets into something really, really challenging before we try to step in. So for anyone you love, early early indications usually are there for us to step in. And I think sometimes we wait too long.
Thank you, Dr. Hurd, for that very human insight and advice. It sounds like connectivity, communication, and maybe substituting, more healthy behaviors or habits for instead of as much as I love potato chips, I gotta let you go, my friend.
Once in a while, I can't let go of my potato chips too much, but yeah, once when I
Once in
yeah,
a while, but I'm overdoing it, like I
yeah,
do need to have that cheat day once in a
yeah,
while. Yeah.
yeah.
So the last thing I wanted to ask was your experience as a female minority innovator in Sweden, in the US, in academia, and also any advice that you can give for upcoming, female and minority innovators that could be beneficial for them.
For me, I'm gonna probably start with the end because I think it goes to my beginning. I think make sure you're in an environment or find people who support you for who you are. And that will give you confidence. Because for me, as a Black girl, as I said earlier, liking science, it was very tough. I didn't really have role models that looked like me. And it was difficult to find or ask for help to navigate. And I think that that's one thing. I think that there are many more programs today for women and minority scientists and innovators. And I think make sure you apply for everything, go to every workshop that you think will be relevant to you. But I remember going to Sweden, I mean, and I was, black in a place where everybody seemed to be tall, blonde hair, and blue eyes, and that, I wasn't supposed to fit in. But actually, the reason I am here today in my position is because of there in Sweden, because it was one of the first places they didn't look at me as black and thinking because I'm black it's something negative in science that I will fail and I won't succeed. And when you have those negative pressures put on you, people don't realize it's much tougher to push them off than in Sweden, where they were like, you're American, so you must be the best. And therefore it was a bar to go up for. And just having that belief that you are supposed to be the best, you can work towards it. So for me, it is the same thing today. Do not stay around negative people. Negative people will not allow you to succeed. And no matter, sometimes you may think that, they are supportive of you, but it's about the actions that they do. And so find the programs, find people who lift you up by telling you the truth. I don't think you should tell people, oh, you're great when they're not. You can be great if you do this. You can be great if you do that. All of us have strengths and all of us have weaknesses. So you need to know what your weaknesses are and work on those. And you need to be in the environment that allows you to see your strengths, build those, to identify your weaknesses and strengthen those. So that to me is, the environment of Sweden really helped me, because the US, it was much tougher. And it still is, even at my level. It's, you still battle um, the implicit biases that sometimes come with being a Black female scientist.
Thanks so much, Dr. Hurd. We've launched a program called I Through Prism that actually provides funding for female and BIPOC inventors in the system, especially the ones that don't yet have funding, because those are the folks that need that support. And so if anybody is interested in that at all, please visit MSIP's website for that. So I think we are at the end of our time. Dr. Hurd, thank you so much for sharing your experiences with us. We're looking forward to hearing about the future of your work, and have a beautiful rest of the summer
You
wherever you
too.
are.
Thank
And
you, Cynthia.
to everybody else also at Mount Sinai, all the innovators and entrepreneurs out there. Till next time, this is Cynthia and Dr. Hurd signing off. Be well, everybody.
Bye. Have a great summer.